Hello everyone, my name is lilyliu. I represent Bio-Byblos, Biomedical, to give you an educational session. Hopefully through this session, you will have a more thorough scope of our 3D cell culture products. The subject of this session is 3D in vitro technology for drug discovery and toxicology. The morphology of cells in animal tissues are three-dimensional. Cells are binding in extracellular matrix. However, the method of cell culture traditionally is in 2D plastic. It is wrong dimensionality. Not only so, cells grown on 2D environment are flat, unnaturally thin, and single layer. Cells in 3D environment are much more similar to cells in a living organism. We compare cells in 2D and in 3D mainly in three aspects: in shape, environment, and behaviors. Cells in 2D are flat with typical thickness of 3 microns. But the cells in 3D have dimension of 10 to 30 micron. It is almost 3 to 10 times more than cells in 2D. In terms of environment, cells in 2D have very small percentage exposed to one another, because 50% of their surfaces exposed to the medium, and the other 50% exposed to the flat culture surfaces. Cells in 3D have almost 100% of their surfaces exposed to one another or matrix. In terms of behaviors, cells in 2D are different from cells in 3D in their differentiation, drug metabolism, expression such as gene and protein, general cell functions, in vivo relevance, morphology, proliferation, response to stimuli, and viability. Now we are coming to cellosponge series. This one is to tell you the structure, appearance, and the size of cellosponge. On the top right, you see one piece of sponge. That is a scaffold with a scale. In the middle is the top view of the sponge. On the bottom is the surface of the sponge. Then now we come to the left side. On the top, we see a cellosponge in 48 well plate. On the bottom, you see aqueous microporosity maintenance of the sponge. On the right is the cross section. This one is 5 days post-seeding of NIH 3T3 in cellosponge plant. In the next few slides, we shall see the applications of cellosponge in plant form. That is cellosponge coating nothing. Like this slide here, we see NIH 3T3 culturing again. On the left chart, cell proliferation determined by allama blue acid in y-axis, along with the culturing days in x-axis. The growth of the cells until day 8 is increasing in a stable path. After day 8, due to our controlling of the size of the spheroid, cell proliferation maintains roughly the same. On the right, we see live and dead standing after 4 weeks of cultivation. Living cells in green and dead in red. This picture we see after 4 weeks of cultivation, there are very few dead cells, which are red dots. This is one of remarkable characteristics of our products. This one continues to show the application of cellosponge. It is NIH 3T3 3D cell culture for day 1 on the top and culture for 5 days on the bottom. On your left are low mag, on your right are high mag. More application of cellosponge. This one is the in vivo study for biocompatibility of cellosponge by 1 to 12 week subcutaneous implantation in mice. In these photos, we see Manson trichrome staining of 2 scaffolds and their surrounding tissues. On your left are the low mag with scale bar 100 micron. On the right are the high mag with 20 micron. Collagen in blue, cytoplasm, muscle, and erythrocytes in red, and nuclei in black. The star side is sponge pores. We didn't see much red and black until week 12. That is to say, the mice did not have much immune response. It shows the high biocompatibility of cellosponge. Not only having cellosponge plant, we also can conjugate some substances, such as collagen on cellosponge. The picture on the bottom shows human foreskin fibroblast, HFF, in both cellosponge and cellosponge coating collagen. Both of them form spheroids, but on the right, spheroids connect, or you say communicate, to one another. That is to say, HFF in the spreading morphology due to enhanced cell matrix interaction with improved cell viability. We continue with cellosponge collagen. These pictures are neurodifferentiation of human MSC on cellosponge collagen. From the left to the right, human MSC in neurodifferentiation median for 2 days, 7 days, and 14 days. The red arrow, the cell body. The green arrow, neurite. Cellosponge coating collagen is stem cell differentiation platform. The figure on the left, you see cell proliferation of human MSC in cellosponge collagen at different time points. The number of human MSC cultivated in cellosponge collagen increased by more than 14-fold after 7 days. The photos on the right, human MSC in neurodifferentiation median for 2 days, 7 days, and 14 days. In the photos of 14 days, we see that human MSC could be induced to differentiate mature neuron or glial cells. Cells in 2Ds usually takes a month to do so, but in cellosponge collagen only take 2 weeks. It almost doubled the time to differentiate. So we can see cells in 3D exposed to other cells and environment almost 100% can differentiate a lot faster than that of in 2Ds. In the Journal of Trends in Biotechnology, we see the efforts to make artificial tissues in vitro for drug testing in liver cells. It also tells us the importance of 3D primary hypothesized culture. The advantages of in vitro 3D models for liver toxicity study. 3D systems allow for enhanced preservation of complex in vivo cell phenotypes due to compactness of cells compared to conventional 2D cultures. Furthermore, tight cell-to-cell junctions in 3D systems help maintain cell viability and high-level liver-specific functions, such as cytochrome P450 activity, albumin secretion, and urine synthesis. Now we come to 3D primary red hepatocyte culture. A and B on the top show two liver-specific functions, albumin secretion and urine synthesis. Black bars are primary red hepatocyte cultured in cellosponge. White bars are the same cells in collagen-coated surfaces. We can see that these two functions of hepatocyte cultured in cellosponge are much higher than those cultured as 2D monolayer on collagen-coated surfaces. On the bottom, you see two pictures of hepatocyte culture for 7 days in low and high mag respectively. Cellosponge not only can conjugate collagen for stem cells, but also galactose for hepatocyte cells. On the top, you can see hepatocyte culture in cellosponge galactose form spheral in 1 day of seeding. The most right, the arrow in the figure shows spheral attached on sponge surfaces. On the bottom, we can see hepatocyte culture in cellosponge galactose are in a very good shape. Only 1 day of seeding has few red dots. In day 3, 5, and 7, the hepatocyte culture in cellosponge galactose are all living. It is because we can control the size of the spheral within 200 micron in order to avoid hypoxia of the cells in the center of the spheroids. This slide shows us the image of hepatocyte culture from 1 to 12 hours after seeding. We can see the hepatocyte form spheral after 7 hours of seeding. We continue to see the application of cellosponge coating galactose. In the pictures here, we see the comparison of 3 different platforms to culture primary red hepatocyte cells. From the left to the right, collagen monolayer, PET galactose-elated membrane. On the right is our cellosponge coating galactose. Hepatocyte culture on collagen monolayer were relatively flat and spread on day 3 onwards, which means rapid loss of differentiated functions. Hepatocyte spheral formation on PET galactose-elated membrane took 3 days to form and often collided with spherules nearby to form larger ones. In cellosponge coating galactose, hepatocyte immediately organized into 3D spheral in 1 day of culture and maintained similar sizes until day 6. It also means hepatocyte cells in the center of the spheral won't die due to hypoxia. Furthermore, hepatocyte spherules formed in cellosponge galactose were smaller than those formed in PET galactose-elated membrane. More application of cellosponge galactose. On the left, albumin secretion and urine synthesis functions of hepatocytes in cellosponge galactose in black bars and collagen sandwich in white bars. We can see the black bars are all outperform those of white bars in day 1, 3, 5, and 7. On the right, we see immunofluorescence of mature hepatocyte markers, staining F-actin in red, E-cadherin in green, MRP2 in red, and CD147 in green in hepatocyte spherules 48 hours post-seeding. Through F-actin, we see a clear cell structure of hepatocyte spherule in red. In E-cadherin staining, it shows us cell-to-cell interconnection, and also the maintenance of cell viability during long-term culturing in green. MRP2 staining showing us the apical domains of the hepatocytes in red. This tells us that transport protein within healthy living hepatocyte cells. We didn't see much red in hepatocyte pictures of collagen sandwich. Lastly, CD147 staining marks basolateral layer in green, showing us this hepatocyte cultured are not transforming into other kind of cells, and they are not dying. On the top, FDA staining in the hepatocyte spheroid show us an accumulation in the biocanalicculus between two cells. On the most left, the formation of biocanalicculus of hepatocyte cells in collagen sandwich. It took 48 hours after seeding. The rest of the picture are that of cellosponge galatos, starting from 16 hours post-seeding, 24 hours and 48 hours. It is a lot faster than that of reported in collagen sandwich, which normally occurs between 48 hours to 72 hours post-seeding. The picture on the lower left is the subcellular microstructures located inside of the hepatocyte spherule. The spherule is cultured for 48 hours. It is to demonstrate a space between neighboring cells, the biocanalicculus, with presence of microvilli. This is basin-level gene expression of CYP450 enzymes. The gene expression of CYP450 of hepatocyte in collagen sandwich will assume 1 in y-axis. We see on day 3 hepatocytes show higher expression of all in cellosponge galatos than hepatocyte culture in collagen sandwich, except for CY2E1. But on day 5, the spheral culture in cellosponge galatos show a 3.42-fold higher expression of CYP2E1 than that in collagen sandwich. On the bottom, in Table 1, you see two different kinds of inducer drugs. They are standard drugs used by BD to test drug response of hepatocyte cells. On the top, you see two cytochrome P450s, CYP1A2 and CYP2B2 in the drug induction experiment. After induced drugs, cellosponge galatos culture hepatocyte spherules shows higher metabolite production than that of the collagen sandwich. As a conclusion, cellosponge series products can be used in general soft tissue 3D culture, sponge for in vivo study, stem cell differentiation platforms, 3D primary cell culture model, and lastly drug and antiviral testing. Human body is not a homogeneous entity and is composed of different kinds of cells. Different cells responding to different mechanical stiffness. The mechanical stiffness of cellosponge series is smaller than 10 kPa, so it is good for some degree of muscle cells, brain cells. In terms of Go matrix, its mechanical stiffness is greater than 120 kPa, so it is good for connective tissue, bone cells, etc. The following is another product, Go matrix, It is hydrogel-like 3D scaffold made of gelatin. The primary characteristic of Go Matrix is its uniform pore size. Our standard pore size is 130 micron. There are also 60, 90, and 200 micron pore size available presently. On the left is the top view of Go Matrix. On the top right, the side view, showing us the array of uniform pores. Here are the cell types Go Matrix is designed for: epithelial cells, muscle cells, and connective tissue. Almost all the organs are composed by these few kinds of cells, except hepatocyte and lung. These cell lines are also most of labs use. Go Matrix has been tested with MDCK, C2C12 myoblast, and free T3 fibroblast. Here's an example of myoblast cells, C2C12, culture in Go Matrix. After only 1 day of seeding, C2C12 stretched into lines. By forming strong contacts with cells in neighboring pores. Showing the pores of Go Matrix are round, but can culture those elongated cells in morphology, because they are interconnected. On the top right, you can see the morphology of C2C12 clearly. On the left, showing us that our muscle is composed of many myocubes. On the bottom right, C2C12 was observed with thick, fused myocubes with multiple nuclei in blue, after cultured with differentiation medium for a month. This phenomenon cannot be observed in 2D cell culture. Within 1 day, MDCK cells form spheres of cells that joined into both closed structures and open interconnected structure. That is, closed and open cysts at the pores and the aggregates. In the picture, you can see apical membranes in green, using antibody against GP135. The red part are basolateral membrane, normally face neighboring cells and substrate, and in contact with other cells and substrate. Nuclei in blue. You cannot see this in 2D cell culture. Only 3D cell culture can produce such functional cells. In this case is MDCK differentiates into multicellular structure of polarized cells. As a conclusion, we can see cell sponge is good for cancer cell lines, such as MCF7, MDA, MDB, mouse fibroblasts, NIH3T3, human foreskin fibroblast, HFF. Its applications is soft matrix for 3D cell culture and 3D tumor model. In terms of cell sponge gelatos, it is designed for primary rat and human hepatocytes and hepatocyte cell lines. Its applications: in vitro drug testing, 3D cell culture model for hepatitis virus infection and replication study, hepatocyte-like cells maturation in differentiation study. In terms of cell sponge collagen, it is designed for mouse and human embryonic stem cells and human mesenchymal stem cells and cardiomyocytes. Its applications: cardiomyocytes long-term 3D culture, mouse and human embryonic stem cell and human mesenchymal stem cell differentiation. Lastly, gold matrix. Gold matrix is good for epithelial cells, muscle cells, and connective tissues. Its application, long-term 3D culture, and because of its custom and uniform pore size, it is good for observation of cells morphology and proliferation. Here we give you a rough 3D cell culture comparison. There are few leading types of 3D cell culture technologies, mainly in two categories, hydrogel and scaffolds. In the format of scaffolds, there are two kinds of material found. First is synthetic sources, such as PS, PCl. The problem of this kind is, technically speaking, the pore size cannot be made smaller than 200 micron. Secondly, natural sources, such as HPC scaffolds, which is our Cellosponge series. Our Cellosponge series can control the pore size within 200 micron to avoid the hypoxia of cells in the center of the spheroids. There are the third kind of 3D cell culture format between hydrogel and scaffold. It is hydrogel made scaffold. Our gold matrix, the gelatin scaffold, is this kind. I represent Bio-Byblos Biomedical to thank you for your time. For more info, please visit our website www.bbbmd.com or email us with sales@bbbmd.com.tw or lilyliu.bbbmd@gmail.com.